so the goal of this workshop is going to be get everybody started and doing something. So the first thing that I want to talk about is just how to sign up and make sure that everybody does not leave here before you are signed up. So what you can do is go to this link right here, sciencecoach.t-bio.info. And if you have not already, you just create an account. And I'm going to show you how to access the program so that you're registered to be a part of the program. So once you're logged in, you'll see something like this. And what you'll see here is this is your points. And this is kind of like how you get to a level that you can actually use the platform for your research. You have to accumulate at least 3,000 points. So for every activity that you do, and I'll show you some of the activities today, you'll get a certain number of points. The other thing is that it would be really great for you to complete your profile. So if you click on this thing right here, if you go right here, you'll see your profile, and then there you can add like your picture and some information about you. So one thing that we do want to know is your school, or maybe Science Coach as an organization or something like that. And another thing that once you're here, you can go over to programs, and you don't have to fill this information out right now. Just saying that it's there would be nice to have that. So right here under programs, you have a drop down that says pilot program. So that's the pilot program that we are gonna do. And when you go there, there's just a button that says to join that program. So it looks like this. And for you, it's going to be a button that says something like register, right? Okay, so once you've done that, you're, you will see this badge. Yeah, so just that register thing. You will see this badge right here, right? And so that means that you're a part of the program. For anyone that will be joining maybe at a later time, just walk them through this process, right? So register and join this program. And this is what a complete profile would look like, right? So you can see this is the person, that's his name, and then you see several things right here. For example, you see that he's got level 3. So level 3 means that he's completed a certain number of basic courses. And he's got 30,000 points. So this person is already doing some research projects on their own, and he's an expert, and you can see that he's taken the courses, and when it says completed, that means he's completed them, and this shows their progress in a specific course. And this is a part of the program that he's in called Omics Logic Genomics. So a little background about this site, the main site, is used by over 2,500 people, mostly graduate and research, and some now are starting to be undergraduate. And like Jill said, this is the first time we're doing it with high schools that are going to try and actually do their independent project. We have another program that they're just learning the basics. So if you go under the pilot program page, there is all that information is in there. And I'm just saying this because it can be changed. So starting from 7th of October, 14th October, 21st, and then if you keep scrolling, 11th of November. So these are the dates of something that will happen, like an event. You can also go to this curriculum tab, and that will tell you kind of like more details about each meeting, what we are going to try and cover. Okay, so this is today, in-person meeting at BioSTL. And then these are the things that we're going to go through: processing next-generation sequencing data, statistical analysis, biological interpretation, project submission workshop, and then submit topic for research. So these are the scheduled sessions. And today we're going to try and look at kind of a high-level overview of some of the topics that we'll go into deeper later on. All of this material is also supported by the online independent courses that you can take. But the goal will be to complete Transcriptomics 1, 2, and 3, which is their own version right here. I just want to kind of explain how this works. So once you take that course, you'll see here the outline of the topics. once you get to the bottom of the topic, there will be a button that says complete or finish or something like that, and you have to finish that to get the points. So I was mentioning those points, and this is kind of like a part of what we are trying to do, is we want to figure out a way so that we know that you're ready to do things independently, and we can help address any kind of gaps that we see from the standpoint of the curriculum, what it covers, and the exemplar projects that we'll cover. Okay, so the idea of this, and this is just a schematic that explains the logic of what we are trying to do. What we're trying to do is we're trying to introduce a topic. So let's say next generation sequencing would be a topic, or transcriptomics would be a topic. And we will, in the context of that topic, introduce a couple of projects. And that might be one project or two or three sometimes, depending on what we are talking about. We'll talk about each project in the context of what type of data. For example, how many samples, what is the condition, what is the control, and what are we trying to analyze? Why are we looking at this problem? Then, based on the data that you put in, you will get a suggested pipeline. So basically, we will tell you this is how you might address this, but you'll also have some variations. So you can make those decisions on your own. And eventually, as you get through several of these examples, we'll do an assessment, so we'll make sure that everybody understands what they're doing, and then you're ready to apply more complex things and work on this independently. So in terms of how data is connecting all of these different aspects, I want to just briefly talk about the different types of projects that we will try and cover. Obviously, there's a lot of interest in molecular data in precision medicine. And different types of data are going to be important for different aspects of precision medicine. There's also, for example, projects that deal with biotechnology. In what sense? There's a project on there that talks about a cell line that is used for production of biologics. So, when you produce vaccines, for example, you need to have some medium to kind of produce that. And so a cell line is typically taken from an insect. Now, if you take that insect, insects have their own viruses, and they can get contaminated. So it's important from the FDA standpoint to check those cell lines so that they are not contaminated with any other types of viruses. The question is, how do you check for that? So there's a whole lab dedicated to this at FDA that actually regularly samples batches of these different vaccines and will test them whether there's some presence of other types of viruses. And an important aspect of it is that a virus genome is fairly short compared to, let's say, human genome. So you have to have many, many of these genomes and try and test whether a portion and whether a significant portion of that genome is present. So we'll talk about projects that range from biotechnology to precision medicine, more on the clinical side, or the biomedical research side, and agriculture. So in agriculture, it's also important, for example, to understand traits. How are these traits that we associate with the number of, let's say, tomatoes that grow on a single plant, or how red they are, or other things like drought that might or might not affect a certain type of a crop. So analyzing all these different biological, essentially, questions really relies on this kind of like an idea that you have something on the surface. And in the case of clinical projects, we're talking about clinical data, right? So clinical data is what stands out from a superficial examination. So a patient comes in, you kind of take these superficial, for example, a person is overweight or a person is of a certain age, right? And we call those phenotypes. And then underneath all of those phenotypes, we have this different level of molecular data that essentially can sometimes explain on a more precise level and a more individualized level what is really going on. Now, of course, there is a big problem of noise here because you're capturing what is happening in every living cell. So just because a cell is living in a little bit of a different way doesn't mean that something wrong is going on, right? So separating that signal from noise is a critical component. Now, the first project that I want to talk about is a project that relates to precision medicine. So before we talk about precision medicine, let's try and understand the relevance of precision medicine and why molecular data, right? So I mean, precision means that it's precise, right? And so what happened over the years that we are concerned with precision? So over on this side, this is a population of patients that were compared to other patients, to a collection of data called the Cancer Genome Atlas. And what you can see here is expression of a certain gene and an expression of another gene. So you have two axes. You have here expression of ESR1 gene, and here's ERBB2 gene. So HER2 is the protein, and ERBB2 is the gene that encodes that protein. And the reason why there is a test for HER2 and, like you're saying, mutations in HER2 are important is because of Herceptin. Now, Herceptin is an antibody, so it essentially blocks a receptor on the cell. So what happens is, this is kind of schematic that explains what happens. It's related to breast cancer, and those cells that have that receptor, which is responsible for growth, this proliferation of growth that we associate with tumor growth, is the key, the gateway for that whole pathway. So if you block that pathway, it doesn't grow anymore, right? So it's a very effective, precise treatment because it will only affect those cells that are out of control. They're just growing too fast. So, but because this is a very small, obviously, receptor, it's a protein, any change in this gene can modify the shape, and this specific antibody is not going to work. So if we find a mutation, and that mutation is going to be changing the structure of the protein or preventing that protein from functioning well, that specific drug is just not going to work. On the other hand, if we can identify that this protein is there in the right form, then we know for sure that that patient is going to respond to treatment, right? So now this kind of puts together the idea of personalized medicine, right? So there's two kind of concepts here: precision medicine. And why precision? Because we're looking at a molecular level, at a very small level, to understand exactly what's happening at that level. The personalized aspect of it is the idea that we can take a population right here, and we can use molecular profiling to stratify these patients, or even look at individuals instead of large groups of patients, right? So if we can look at prognostic markers, for example, markers that tell us how far away from today is this person likely to die from this disease or have a recurring event. If we have, for example, drug sensitivity and resistance, these are living organisms, so obviously humans, but we are talking about changes that occur as a result of external pressures. So when we give chemotherapy, which is very toxic, the response to that could change the DNA and could lead to all kinds of other changes. So it's really important to not only do it once, but continue to do it over time. And then also to look at adverse events. So a lot of people that take one type of medication take another type of medication, and considering all those factors together, really what we're talking about is developing a plan, a treatment plan, right? So we talked about how it's important to diagnose and kind of separate people into groups. Then it's important to understand how they change over time and monitor that process. And then eventually it's important to really have a link to something that is effective at this point in time for this particular patient. And that's the concept of precision personalized medicine. Now, one aspect of this is that obviously you cannot try a lot of drugs on people, right? So the problem becomes, well, how do you know? You know, you have thousands of compounds. How do you know how to look for something that's effective? So what this shows is actually inside each one of these holes is a bunch of cells that are going to be typically cancer cells, and then here you just drip a bunch of drugs, right? So you try, this is called high throughput testing. So you take a lot of different compounds, a lot of cells that resemble what happens in a patient, and you test what the response might be. So those are two different types of data, right? This is data that's coming from the organism, right? And this is data that is telling us something about drugs. So there are multiple components that this model is going to be lacking, but there's a lot more that you can do there, right? And this is going to maybe give you a lot of potential information, but also you don't collect that much data on them, right? So there's benefits and downsides to all of this. Now, how does bioinformatics fit into all of this? So bioinformatics is really a combination of biology, statistics, and computer science. And the combination is really in the fact that we have, so bioinformatics, information technologies, right? We need to assemble a lot of different data to start applying any kind of algorithms to it. And that's one part of bioinformatics that just deals with collecting a bunch of different data into databases that people can use. And we'll learn a little bit about some of those databases that are available where you can find ideas for your projects. The computer science component of it is obvious. You have to deal with large data sets, but what else do you think? Why computer science? The computer science refers to the algorithms, right? Because when you're talking about detecting patterns, that's really statistics, right? Statistics and statistical methods can help us identify patterns and know that those patterns are not noise, they're not random. But why do we still need computers, though? The algorithm is trying to explain a question that I understand, right, to a computer that's not able to understand what I'm trying to do, right, and break it down into steps that together produce the result that we would like, right? So an algorithm is an important part of this because essentially from the data that we collect to something that we can know how to interpret, there are actually many different steps that you have to go through. And a lot of them have to rely on parameters that, yes, we could do as humans, but because there's a lot of these steps, and every time you have to adapt the algorithm to whatever the input is, the algorithm part of it is really what achieves what you were talking about, right? Saving that time. So if we were to combine these different things, we could focus on one of them, right? So we could learn about the biology. We could talk about precision medicine from the standpoint of proteins and how they interact, right? We can talk about the algorithms, how to write a program that does certain things, right? Or we could talk about statistical methods that help us detect patterns. But together, that's what really makes bioinformatics unique. And in some sense, each one of those is a completely separate discipline. So you might decide to go into any one of those disciplines eventually, but in this course we're going to focus on the logic of bioinformatics. How do we approach a project, and how do we utilize some of these things with some level of understanding so that they work for us? Okay, so let's talk about one of these bioinformatics projects. Let's start from the data. Where can we find data? So obviously, NCBI, now if you have your computer, why don't we go to NCBI? If we go to SRA, and I'll maybe do this together with you. Okay, so right here in the drop-down, you can see all the different databases, and like that, you go to SRA. Now, inside this field right here, you can search for whatever you want. So, you know, search for, I don't know, lung cancer, and, you know, somebody else can search for, I don't know, potato, and somebody else can search for something else. And I'll search for breast cancer. Okay, so now you're seeing how you can find this raw, publicly available data, and let me just explain briefly how it gets here and why it gets here. Like, why would somebody sequence a bunch of data and submit it to NCBI? So it's a requirement of grants. When you submit a grant and you submit a publication, you have to reference where's the source data. Does anyone know what are kind of like the main components of a paper? Hypothesis, literature review, abstract. Okay, so the methods are going to describe what data was collected and how it was collected, right? What was done to the data and what did the data show? So a lot of the methods is also going to talk about, why they did what they did, right? So they're going to start from hypothesis, or somebody here mentioned hypothesis, don't remember who, right? And they probably collected the data specifically to test that hypothesis, right? Now, that's them, right? That's how the data got here. So we can read about each one of these and we can understand, well, why was this data generated? And it's important to understand because if you wanted to test, a specific subtype of cancer, and I'm just assuming that this is, just general kind of gene expression, I'm going to be wrong. So I do need to understand why this data was generated. But I don't have to already have a hypothesis, right? And so there's kind of these two different worlds. There's a hypothesis that we want to test, and we have to have a very specific idea, and we have to kind of know how to test it. But the other world of it is called data mining. So we don't really have a hypothesis. It's what is typically frowned upon in publications when you do these fishing trips and you're just, whatever comes out. But that's exactly what we're going to start with, because that is going to help us come up with a hypothesis, right? And so because we're not experts here in any particular topic of science, it's difficult for us to come up with a good scientific question. If we look at the data and we spot some trend, and that trend starts to be interesting, now we can start looking at why it's interesting, and that will help us create that hypothesis. Now, I want you to know that, this is just one part of this, but let's briefly kind of understand what do we know about these things. So these are just actual files, right? So this is just a reference to the file. Again, my mouse is tiny here, but it tells you right away kind of what it was generated with. Illumina HiSeq 3000, it tells you kind of what is the size of that data set. And what it also will tell you is each one of these actually belongs to a bioproject. And a bioproject is actually a collection of these samples. So you might have 25 samples in a project that will be called a bioproject. And then you can read what is this bioproject. So for example, if we go over here, it will tell us, okay, metastatic behavior varies significantly among breast cancers. And you can read that abstract that you talked about. And then here it talks about what is the data type, transcriptome or gene expression, and then, what is the organism that this is from? Mouse, exactly, right. So mus musculus right there is mouse. And then how many data sets do we have here? 10, exactly. So we have 10 samples and 10 SRA experiments. So those would be the files that we're looking for. So if I want to get this data set, what I would do is I would click on this 10, and I will have the list of all of those files so I can actually see what did they try to do with the data, right? So here now you can see kind of the conditions that they were studying, right? So monocytes, neutrophils, etc. So now you can start understanding how what they were trying to understand or what their conclusion was is related to data. So that's number one, right? So number one, we can use that to refine our search. Obviously, if we want to look at mouse, if we want to look at cell lines, if we want to look at patients, those are different levels, and we can define it in the search. And we can also look at different types of data. So let's take one of these projects as an example and try to understand what they tried to do, why they were trying to do it, and then link it to the data that we can find here on NCBI. So the mammogram shows this growth right here, and so that is breast cancer growth. And one way to characterize this growth is by linking it to a specific tissue, right? So just by looking at these different tissues, it's kind of important because you will read about these different subtypes of breast cancer, and they're typically linked to tissue. Now, how do we know whether in this tissue there are specific proteins that could be helpful in our diagnosis? So this is staining. Yes, immunohistochemistry, which basically, if you see right here, these are the different proteins that we are staining for, like EGFR is probably familiar. And these are the different subtypes. So this is triple negative breast cancer, HER2 positive, luminal A, luminal B. So knowing these proteins, their presence or absence, so essentially the stain is something that connects to that protein but also has a color. So if the color comes out, we know it's there. So we look at it and we know just for a single protein. So what is the limitation of a test like this, do you think? So first of all, you can. Theoretically, you can come up with 20 different types of antibodies that will connect to that protein and make it a stain, and then you will do it, but you'll run out of tissue. So you cannot take continuous biopsies, and therefore, and also the other side of it is that what if that protein has changed, right? So now it's not going to connect, you're going to think it's not there. So it really complicates that. And so that's why we turn to gene expression. And this also helps us connect it to these subtypes. So basically, if you see this pattern corresponds with a subtype. Now, does anyone know what is this here on the side? So a taxonomic tree uses it. It's called the dendrogram. A dendrogram shows connections, right? And so the height of this dendrogram shows distance, how closely related they are. So this is a dendrogram that serves two purposes. One, it helps organize the data. So essentially, it's a clustering algorithm that will make this into most, so the ones that are close together means that they're similar to each other. So we'll organize them by kind of similarity. And then, like we said, the color is what actually helps us right away spot some kind of a pattern. So we actually use statistics, which is what fold change would kind of show us only the genes that are differentially expressed, help us understand how much of the difference exists, and then we can kind of look at it and know what's going on, right? So this is, so you mentioned microarray, this is called a panel called PAM50 panel, and PAM50 stands for 50 genes. So these 50 genes are genes that were selected to help us understand some kind of information. What kind of information do you think we can get from this kind of a test? So here's a comparison of two different panels, PAM50 and Oncotype DX. So these are actual clinical tests that are used in the clinic for breast cancer patients. So I'll kind of read them. So here you have distant recurrence percentage. Here you have follow-up time in years. And you have three different categories. So three different colors, blue, gray, and yellow. So this is the 100% of a clinical study. So you have, let's say, 30,000 patients, and you observe them for 10 years. And you're looking at the number of patients that have, so this says ROR, which stands rate of recurrence, and this is RS, so this is rate of survival. So you're looking at how many patients die at each point or they have a recurrence. So now, why three? You actually have three categories of risk. Those patients that tend to die quickly or have a rate of recurrence, and you can see that those lines are actually very close to each other, right? So rate of recurrence and rate of survival is a correlated factor. So you can see that both of these, right, these are two different panels, so they use two different sets of genes. One uses 50 genes, another one uses 35 genes, so there are different types of panels. Like, which one's better, do you think? What I would say is better is the PAM50 because there's a more clear difference between the three different groups, right? So if there's more difference, that means it will be easier for us to spot the difference, right? And the other thing is that it is more clear early on. So early on, I will be able to predict what will happen 5, 10 years from now, which is in itself amazing, right? That I was able to select genes that will tell me what will happen 5, 10 years from now, just by looking at their expression. So this is called the Kaplan-Meier curve, and it typically is used for survival and recurrence in these different clinical studies. Now, why is this information about risk actually useful? It's useful because now we can select the right treatment. For example, if somebody is in a high risk, we can use more aggressive combination of treatments because it's more important to prevent death than actually think about side effects. If somebody's in a low risk category, maybe we will be more thoughtful about trying something and seeing whether it works, and also minimizing the side effects. So a lot of times the recurrence actually comes because you pump all these medications and radiotherapy, etc. So another aspect of it is we really have three major methods here to talk about, right? We have surgery, which is not going to be, for example, effective if a person has metastasis and the cancer is spreading beyond a location that you can actually capture. Radiotherapy, a lot of times, is used even before surgery to minimize that growth and maybe even shrink the tumor to cut it out. And then obviously chemotherapy, which chemotherapy is actually very, very toxic, right? So if we could avoid chemotherapy, that would be good, right? And so that's why we talked about this targeted cancer therapy, right? And that's one of the things that we see a lot more today, is that we are trying to find such a treatment that will only affect a certain type of cells, right? So if we could design a treatment that will only kill the cancer cells, right, that would be perfect. But in the case of Herceptin, of course it was, tremendous progress that all of a sudden this drug is available, but there's not many of those, right? So there's not so many of these targeted therapies. And with Herceptin, we actually know the process. So it was an understanding that came later on. First, the drug was discovered, and then later on, people understood exactly how it works. But now we understand the whole signaling pathway and a whole set of genes that is critical to stop growth. Now, because we know the signaling pathway, why do you think it's the protein on the surface of the cell that made it such an effective treatment? So the deeper it is, right, the more difficult it's to get there, and there's all kinds of processes that can stop it along the way, right? So it's important to understand the biology to kind of understand how these things work, and also it's important to think of this in terms of pathways, because really there's multiple players in any single event, and we need to typically select the most easily accessible and the one that is a critical player. And that has to do with data. So ultimately, we would like to, by looking at data, understand what's happening inside the cell and then make a decision kind of like this. So now we're going to take a real paper and we're going to take some data from that paper and try and see if we can do something like this. So here's a paper. It's called Modeling Precision Treatment of Breast Cancer. You can see the link right here at the bottom. And in fact, I think if you look for this on Google, you'll probably find it. So the name of the author is Diamond, and it was published in 2013. What it is they collected patient-derived cell lines from breast cancer patients, generated molecular data that gave us information about mutations, copy number variation, methylation, etc. And they integrated this data together to build these predictive biomarkers that will tell how maybe they could be separated into different subtypes. The other data set was to try and put drugs on these cell lines and see how effective these different treatments are. So they took over 70 different compounds, both the ones that are approved and in clinical trials, and they tried how effective they are. Now, how can we measure effectiveness of a cancer drug on cell lines? Does somebody know what is the actual data that comes out of that high throughput experiment? So in this case, they used inhibition of growth 50, which was how fast, so time, until 50% of the cells were dead. So that's what they did here. And so we have a bunch of essentially data sets that we can work with now: genomic data, transcriptomic data, IG50 data, so a bunch of different data. But what is common between these data sets? Number of cells. So the number of cells is exactly the same for all your molecular data. It changes here. So look at this table right here. This says gene 1, 2, 3, 4, sample 1, sample 2, sample 3, sample 4. Which one of this is going to be consistent across all of these data sets? Okay, what's the same are going to be samples, right? Because those are the objects. So for example, we'll have luminal subtype, and that will have 10 samples. And so those 10 samples are going to be exactly the same for every single type of data. Now in data science language, these are called objects, and these are called features. Features are characteristics like time to death, gene expression like you're seeing right here, or which positions are changed or not changed, right? Those are all features. They help us characterize the data set. But for us to work with this data set, we're looking at defining these samples using these different characteristics, right? So if we take an example from agriculture, if we have 10 oranges and 10 apples, and we want to compare those two using a variety of different features. We can use, for example, color, right? That's a feature. We can use taste, we can use smell, we can use all these different characteristics, and we can measure them in different ways. And so, but ultimately, what are we trying to study? Difference between apples and oranges, right? And so those have to be present in all of our data sets. So that is what's common between them. Now, I would like to ask you to read this paper so that we can discuss it further in future sessions, right? So what you're trying to understand here is, again, those things that we talked about before, right? So what are some of the things that we discussed about a paper? What's in a paper? The abstract, the methods. What else? The hypothesis. What else? What? Yes, background. There's also the name of the journal and the date. There's a lot of interesting things here. But basically, yes, there is the abstract right here. So at least try to work through the abstract, and that summarizes what is going on. And then look at the methods, right? So the methods is what we're going to be talking about later. Now, all of this data is accessible on NCBI, and that's why we're using it, right? We're not stealing anybody's data. This is publicly available data. And this data is patient-derived cell lines. So patient-derived cell lines doesn't mean that they took a patient and took some cells from them and grew them. What it means is that there are these cell lines that will live forever, and they're in all kinds of different banks, and you can just buy them. And you buy them, and this could be from a patient that died, 50 years ago. But then you can do all kinds of experiments. But just to look at, some of this information that we have on NCBI, you can see that this is a bio project, right? And so this is going to be one bio project that collects all of this data. And that's your assignment, is to find this data on NCBI. So for next time, you have to read the paper. It doesn't matter if you don't understand it, just read it. But you have to find the data, okay? Okay, so let's just take one of these data sets, right? There's many data sets here, but let's just focus on next generation sequencing in the context of transcriptomics. And let's think about how did this data, how is it that we have this data, and what type of data is this, and kind of why. So first of all, what I wanted to do is briefly introduce genomic data so that we all understand kind of what is happening and why is there so much of this data available. So back in the 1950s, the DNA structure was discovered, but what that means is that really we started to understand that the sequence has, that what goes one after the other is really what's important, right? And that's where sequencing comes from, right? But back after that happened, it took a long time before this major effort where the first human genome was assembled. And the first human genome was assembled, cost a ton of money, huge teams. And two things came out of that project. Well, besides the actual human genome, what came out of that are two technologies. One of them is shotgun sequencing, which is what Illumina is based on. And another one is actually the computational power that we can use algorithms and kind of solve these huge computational problems. And because of that, the method of actually knowing where we are starting to sequence is less important, because now we can rely on algorithms to connect these short reads and align them to some big reference genome. So those are the two major breakthroughs that make this data affordable, because we can sequence short reads, and they are not time consuming because we don't have to annotate the whole genome and know from where we are sampling these short reads. We use computers to combine them into longer sequences and map them to a reference genome. So when Illumina developed one of their first sequencers, the breakthrough was that the cost of sequencing started to really go down really fast. And as a result, and you can notice kind of the timeline is not linear, but you can see that 2006 to 2015, we already had a whole 1,000 genomes assembled. And after that, by 2018, actually there are more than 150,000 genomes assembled. And just recently you might have seen that the UK Biobank project is the most ambitious one to date. They want to sequence how many genomes?
I didn't see how many, but it was, I think, in over 300,000?
500,000 genomes, yeah. So they want to assemble 500,000 genomes. And today it's like, okay, I mean, big news, but this just happened organically. So this last portion, you can see there was a consolidated effort to sequence 100,000 genomes by Genomics England. And at the same time, the Broad Institute, just because they did sequencing regularly, they already sequenced 100,000 genomes. So there's a lot of data available. This data is cheaper and cheaper by year. And we now know computationally what to do with that. Now, what does this data look like? This is a sequencer, and it produces something that looks like this. So there are two formats, FASTA and FASTQ, and they produce these sequences of letters. And the sequences of letters could be in longer portions, right, or in shorter portions right here. So right here you can see these are reads that have both the sequence and the quality of that read. And so to understand what that really means, let's take a look at a little bit better what is this. So these are two FASTA files. And the FASTA file just kind of has a continuous sequence of letters. For example, tuberculosis has 4 million base pairs. Does anyone know how many human DNA or genome has? 3 billion. All right, yes, you're the winner. You can have it. So the 3 billion base pairs just as a sequence of letters, right? That's the reference genome. But when we sequence, this is the process of next generation sequencing. So we take the full DNA from a sample that is broken down into short pieces. And once we have these short pieces, they are selected and put in this flow cell. In the flow cell, they're actually amplified. They're amplified so that we have enough material, and then they are placed into the next generation sequencing. So inside, what happens is that this whole machine, all that it does is it looks at every single cell, which inside has just one type of nucleotide, and it measures color. And so by the combination of different colors, it can tell between these different letters. But obviously, because it's color, you cannot be always exactly sure, and that's where the quality comes in. So this is what the read actually breaks down into. You have the label of that read, then you have the sequence of that read, and then down here you have what quality is assigned to that read. So how reliably do we know that that's a letter? Now also, there are two types of ways you can do it. One is called single end, and another one is called paired end. So this is explaining the single end and paired end. You can read, these are directional, that's why the sequence is important, but why would we want to do paired end? Well, this is cDNA, right? So it has two strands, right? And so that's why you can read it from both sides. So reading from both sides, meaning that you're not reading the same thing twice. You're reading from both sides. So if you have a consensus, it's a fairly reliable thing. That's why paired end is a more reliable. You've got quality on both sides, and you can really make assumptions about what is really the best thing. Now, next generation sequencing applies to a variety of different data types. So using the same technology of reading those sequences of letters, we can actually understand something about many different molecular processes. So right here, we have phenomics. And you will see that in a lot of times, in a lot of newer publications, people are talking how in cancer, actually just looking at genomic data is not very informative, right? The morphology of those cells and what they look like is really a lot more informative. So that's phenomics, right? One type of omics. Genomics, so what are we looking for when we talk about genomic data? Who knows? We are looking for similarity, so we're aligning sequences of genes, for example, but not only genes, but we're looking at, these are going to be either substitutions, right, when one letter is substituted by another letter. What other options are there? Deletions, insertions. So that's how we analyze genomic data, is basically we align the sequences and look for one of those three events. Obviously, there could be whole chunks missing, right? If a whole chunk is missing or added, that's not going to be the same kind of a comparison, but ultimately talks about the actual sequence of the DNA. So this is the result of an analysis that compares somatic mutations. So it looks for control, a sample, or like a tumor, a blood sample, and aligns both of those to the reference genome, and then just looks at the differences that happen at the somatic level. Basically, these little balls right here, they are, what are they called? Histones, exactly. And why do we want to understand where these histones are? Well, how tight it is really doesn't tell us anything about expression just yet. What it tells us is how accessible that is, right? And so a mechanism can make it more densely together, and that means it's less accessible, or it can expand it. Now, the DNA, I remember an analogy that somebody said that I think really makes a lot of sense. So if you take all of DNA and you kind of stretch it, and then you put it in the nucleus, that could be something like if you take a rope from New York to San Francisco, and then you put it into a basketball. So it's really, really tightly compressed. And that's why this mechanism of compression is very important here, but also it provides some level of regulation. So epigenetics is a growing field that has been a very important component of us trying to understand what's happening. And ultimately, what it controls is the expression of genes, right? So what happens during transcription? So it goes from DNA to RNA. Somewhere along the way, it becomes mRNA, which is the mature version of a transcript or an isoform, and that eventually gets translated into proteins. So the transcriptomics, when we talk about transcriptomics or RNA-seq, we're looking at a number of genes. So here you see an example of 3 different genes. Anyone know what these things are, these boxes? They're exons, right. So what happens is the splicing out of introns, and what you're left with are different combinations of exons. So you have 2 genes that have all the exons, and then one doesn't have that, right? So if you have a bunch of samples, you can look at a variety of different things. You can look at alternative splicing, which is how many exons and which combinations they exist, or their level of expression. How many do you have? So you can use different types of processing pipelines, essentially, to analyze this kind of data. But when we analyze this kind of data, we're really using a lot of similar methods. We're just applying them in different combinations. So the idea behind a user of bioinformatics is you have a toolbox and you select the right tools to apply to the type of data, and knowing what this data is going to show you. Now this is important because at some point in the process of this pilot, we're going to be talking about projects. And when you have your question, if you can define what it is that you want to find at the end, you will know whether that's possible in the type of data that you have, right? So let me ask you a few questions. Let's say I want to understand the difference between a cancer patient and a normal patient before they have their tumor. So before, let's say, eventually a person is going to become, I know that, right? But I want to understand the difference between these 2 people. One person is going to develop cancer, another person is not going to develop cancer. What should I be looking at? So these would be germline mutations. Germline mutations are mutations that we know by just taking a DNA and comparing to the reference genome. Okay, so what if I am looking for whether a person is going to, be eligible for an immunotherapy? How would I know if that person is going to respond to immunotherapy? What would I be looking for? Protein or gene expression, right? So, I mean, a lot of times they're very correlated, but it's easier to look at gene expression. So I would be looking, and we're talking about next generation sequencing data, right? You can't really detect proteins using that. So I'd be looking for gene expression. What if I'm looking at a neurodegenerative disease, okay? And something that happens over time, a long period of time, and I suspect that it is a result of the environment. So for example, how the person eats, what would I be looking at? Typically, you would be looking for those epigenetic changes, right? So just kind of briefly, what you see here on the screen are some of those techniques. So RNA-seq is for what? transcriptomics. Microarrays are for genomics. Chip sequencing, this stands for chromatin immunoprecipitation. So what it is chromatin are those histones, and you want to find an antibody that binds to those, and that's how you identify that that portion is where the histone is bound to DNA. So you use chromatin immunoprecipitation. Antibodies immunoprecipitation is you just pour them on that DNA, and then what's left are just the regions that have the histone bound to the DNA. So RNA editing are events that have to do with RNA to mRNA, right? So what kind of changes happen? Not exactly, it's a different component, but yeah, it could fall under transcriptomics. So these are some of the different types of analyses that we can do. So let's talk about transcriptomics itself, RNA-seq. How can we use next-generation sequencing? And we'll talk about it in the context of these different things. So if you want to learn about transcriptomics, this is what we're going to be covering in the pilot. You can go from transcriptomics 1 to 2 to 3 to 4. 3 are required, 4 is optional. So 4 deals with single-cell RNA-seq. You don't have to go that far. But transcriptomics 1 deals with just processing. How do I get structured data out of unstructured data? Unstructured data being those short reads. Structured data being, so a table, like when I go from sequences to tables, that's called making it structured. Now I can work with that table. Transcriptomics 2 deals with statistical analysis. If I have 2 conditions, how do I identify statistically significant differences between the 2 using what's called differential expression? So how do I identify those statistically significant differentially expressed genes? Transcriptomics 3 introduces the concept of machine learning. So when we talk about statistical tests, we're really looking at fairly simple, patterns that are easily detectable in some way. There are some properties that are difficult to calculate, and that's why you have a bunch of different statistical tests. But basically, you're looking at something that is upregulated in one condition, downregulated in another. What if you have a lot of different variation? You have, 25 different groups, and they all have their own pattern of expression. Now, that becomes difficult, and that's where machine learning can really help, which is, again, a bunch of algorithms that apply statistical understanding of data so that we can train a model, and then it can help us understand something about what we see. And like I said, transcriptomics 4 talks about single cell, right? So all up until then, we talk about bulk RNA. Bulk RNA means kind of like the average temperature for patients in the whole hospital. What does that really say? Not much. We can't really know what's happening in an individual patient. And single cell separates them using different techniques so we can measure very precisely. The problem is too much information, right? What do we do with all that information? Okay, so now let's do some practical stuff. If you go to server, right here, server.t-bio.info, and you can use these credentials to log in for now, and later on we'll email you your own credentials so we can start tracking what you're doing. So what I'm going to try and do, and hopefully we can get deeper into this in our next lecture, which is going to be dedicated to processing next generation sequencing data. I just want to show you kind of what's going on so you know where to go from here. Okay, so this platform is designed for many different levels. You can go all the way from demo to actual research. You will get somewhere in between, but you will only get access to upload your own data once you're ready for it. So right now we're just going to practice with a simple demo. The demo that we're going to do, so you can see right here that next generation sequencing data. If you want to understand what each section does, you can hover over this or you can hover over this, and you will kind of understand what each section here does. Inside, each one has a consistent interface. So right now what we're going to do is we're not going to go inside, we're just going to press on this drop down. And in the drop down you have these demo projects. So this project that we're talking about with the cell lines for breast cancer is this first one, RNA-seq cell line project. So you can click on there. Each button here represents an algorithm. So this algorithm can be very simple, like a script that loads data and transforms that data into a specific format, like unzips it, or a complicated one that uses advanced methods to calculate something. Essentially, what we are going to do is build pipelines so that we can understand the logic of what we're doing. So how are we going to do this? First of all, you see that the darker button is what you can press. You can't press anything else apart from this dark button. So you got to press on start, right? Here, it's telling you a little bit of the ideas behind these different sections. And let's just think of it in terms of colors, right? So we have these three colors right here. Pre-processing, which is blue, removing PCR duplicates, and removing those adapters. Try to link it back to what we talked about before, the process of next generation sequencing and how that works. Then mapping those short reads on either the reference transcriptome or the reference genome. And then the green is quantification of expression levels. So the first pipeline that you want to build looks like this. Okay, so I'm going to keep this on the screen, and if you have access, why don't you build this pipeline? So you can, to confirm, you have to press on OK. And now you have more options. You have to press on Start first. So let's try and understand what is it that we were trying to do by clicking on these different things. Let me get back there. Okay, so clicking on these buttons actually should make sense. So these are some of the steps that we did, and these are some of the pop-ups that we saw along the way. First of all, we worked on pre-processing that data. Then we did alignment. Then we did quantification. And then there's going to be one of those pipelines, which you will discover later on, is to do differential gene expression. So to understand what we did, we have to understand the file types that we used. So we talked about what a FASTQ file is. So these are the reads. This is the reference genome, right? But that's just the sequence. How do we know where the genes are? The GTF file, right? So the GTF file tells us the beginning and the end, the positions, the beginning and the end positions of each gene. In fact, not each gene, but of each exon. So that's why we can calculate isoforms. So now let's understand what is going on here, right? So this is the central dogma of biology, that we go from DNA to RNA, and then eventually we get to proteins, right? In reality, this is a fairly complicated process. This is from Wikipedia, full disclosure. Don't use Wikipedia for your research project. But what happens is that the DNA, right, we're not looking at the DNA. So even though we're comparing to the genome, we're really looking at mRNA. mRNA is different from just RNA through the process of removing certain elements that are called introns, and they could be combined in a number of different ways. So how exactly does this happen? So let's say we have a gene, and the different colors here are the different exons. In between those exons, we have all kinds of other information that's also sometimes useful, but not expressed. So it's not present in mRNA. So this information, where each one of these positions, beginning and end, is contained in the GTF file. Then we have the FASTQ file, right? And the FASTQ file essentially has a bunch of these reads right here, right? And what we're studying here is we're looking at the isoforms. The isoforms are these different combinations of the different exons. So you can see that this is isoform 1, isoform 2, and those could be differentially expressed. So we are going to look at the abundance of these reads that align to these different regions. So this is the GTF file. You can see here in chromosome 15, there's an exon, there's a position of start and end, and then it tells you the gene ID. So it actually tells you which gene it belongs to, and then it tells you exon number 1, number 2, etc. And what you get at the end is the name of that gene, dot 1, 2, etc. Right? So multiple isoforms per gene based on this combination. Now, when we map our reads, so these are individual reads, right? They map typically within the borders of an exon. But sometimes, first of all, we will get these genes or these reads that don't map anywhere, right? And so some of these are going to have a beginning or an end of one exon and the beginning of the next one. And so what is this? This is actually our evidence that these two are connected, right? So otherwise we wouldn't know that they actually connect. So by looking at these reads, we now know these junctions. Junctions are connections between exons. So we won't talk about this because we don't have time. But let's look at it from a technical review. So here's the sequence. We have gene A, gene B, and gene C. Some of the genes are expressed. They will produce transcripts. Some of the genes are not expressed, right? And then other ones will be expressed at a different level. After this RNA is produced, we break it down into these short reads. And these short reads, we're actually adding adapters. So after they're broken down, we have to add adapters to the beginning and the end of each read. And then they're amplified. So PCR amplification produces much more of the same reads. And then we analyze what is the sequence of each one of these reads with adapters. And so that's why what we first need to do is we want to pre-process. We want to remove the adapters and reduce this artificial amplification of the number of reads because we don't want to confuse that with the actual biological condition that we're studying. So after we do that and we have these clean reads, we need to map them. But question is, what do we map them to? So we can map them on the full reference genome, right? We have a lot of genes and we have a lot of base pairs. To make it computationally more efficient, we can just forget about all the introns and just say, let's just take the expressed portion, which is under 1%. And now we have a much simpler computational problem, right? So we can map on the set of known transcripts. And those would be called the transcriptome. So we take from the GTF file the positions of beginning and end of all of our exons and forget about the rest. The other alternative is we can actually map on the full genome. And we will find additional new things. For example, sometimes, the slides that I skipped, is that you have all of a sudden these different elements from the introns, right? For example, in specific types of cancer, that's what happens, right? And that tells us already that something wrong is going on. But then, as you saw, these reads don't align really well to a defined element like an exon. Sometimes there's overlaps. Sometimes in an isoform that has 3 or 4 different exons, there's different levels. So how do you quantify an expression of something that has multiple levels in it, right? So it's a fairly complex tool that looks at the expression and seeks to define expression of a gene. That's where we're going to end for now. And in our next session, we're going to pick it up from here and talk about how can we modify the pipeline that we just did, which you can actually do from home. You have this, until I get back to New Orleans, so you have this account. And you can just, in the demo, don't try and upload anything because we'll talk about that later. But when you have the demo, so if you just click right here on Create Demo RNA-seq, and you will see that there's a darker color on the buttons that you can click. There's not a darker color on a button that you can't click. And you can try and recreate several of these pipelines and look at, try and understand what does it actually do. So that's all I have for today. We can continue next time online. We will confirm the dates and the time with all of you, and then we'll email you also. I know some of you didn't get the access to your accounts, but we will email you that. And please fill out your profile. If you did get your information, fill out your profile so we know who's who. Thank you very much.